Good morning. Welcome to the seventh session of our biology class 10th class. Hope you are doing fine at home and studying at your home as well. We will be continuing our chapter number 4, that is absorption by roots and rise of water up to xylem. So yes, in the previous class I had explained you the topic. So now we will be moving a bit further. In the previous class I had explained you about osmotic pressure. I had explained you experiments to demonstrate osmosis using cellophane paper. Then I had explained you about diffusion, imbibition, absorption and conduction of minerals in water. We discussed about active transport. We discussed about passive transport. We discussed about tonicity. All these things we had seen. So, and I had given you various questions in relation to the topic that you are supposed to do. Now, one more thing, I have supplied the notes as well as the question answer, and the notes are for your reference. The notes that I have given you, that is for the reference, and the question answer exercises, review questions, you are supposed to write only review questions in your copy. You can refer the notes as well as your book to write the answer which I have asked to write in the copy. Let us move ahead for in the today's session with this chapter 4, that is absorption by roots and rise of water up to the xylem. So the first topic that we will be seeing is turgidity and flaccidity. What is turgidity? What do you understand by the term turgid? When a cell is fully filled with water, it becomes distended. That condition is called as a turgid state. On the contrary, when a cell loses its water, okay, it becomes somewhat loose, and that condition is called as that cell becomes flaccid. Okay, every living plant cell, directly or indirectly, is in contact with fluids. The root hairs in particular are surrounded by soil water. We know that plants which are embedded in the soil, they in the soil itself, they are surrounded by the soil water. All these cells are subjected to osmosis. How will this water enter inside the cell? It will enter by the process of osmosis, and the water continues to enter as long as the cell sap is more concentrated. In the previous class already it has been explained what is the principle behind osmosis. As we know, movement of water molecules takes place from its higher concentration to lower concentration, means where water concentration is more, means from dilute to where water concentration is dilute to where the solute concentration is more. Okay. So due to osmosis, the movement of water will start to take place. As we know that cell sap is having more number of solute, due to which the solution is concentrated. Okay. To enter as long as the cell sap is more concentrated than the surrounding fluids, means from the surrounding, the water will start to enter inside the cells containing the cell sap. Now, when a cell reaches a state when it cannot accommodate any more water, so what will happen? Continuously water will start to move inside the cell, and the cell will become more and more tight. So there will be no place to accommodate new molecules of water. That is, when it becomes fully distended, that condition is called as turgid. The cell is in a turgid condition, is called turgid, and the condition is called turgidity. The cell is called turgid and the condition is called turgidity. When a cell is turgid, its wall is stretched. If you can also take an example of a balloon. When you fill a balloon with air, or when you fill a balloon with water, it becomes tight, isn't it? It becomes stretched. So in the same way, when the cell gets filled with water, the walls become stretched. It is under the pressure. I'll be explaining you what kind of pressure is that. Its wall is stretched under pressure from inside. In its turn, it presses the cell contents towards the center of the cell. So what will happen? on the wall, the the cytoplasm or the cell sap will exert a pressure. Since it is filled with water, it will exert a pressure on the wall. You can understand by this diagram. This is called as Turgor pressure. It will start to give a pressure on the wall, on the cell wall. On the same time, what happens? The wall also exerts an equal pressure. Okay, this is called as wall pressure. So Turgor pressure will equalize with the wall pressure, which will prevent the cell from bursting. Suppose the Turgor pressure increases, then the wall pressure. What will happen? The cell will burst. In turn, it presses the cell contents towards the center of the cell. So the wall will press the contents towards the center. The pressure of the cell content on the cell wall is called Turgor pressure. So the cell content is giving its pressure on the wall, and that is called as Turgor pressure. The pressure of the cell content on the cell wall is called Turgor pressure, and the pressure exerted by the cell wall on the content is called wall pressure. Already I have told you the pressure exerted by the cell content on the cell wall is the Turgor pressure, and the equal pressure exerted by the cell wall inside, means on the cellular components, that is called as the wall pressure. Osmosis also takes place. I have told you osmosis is movement of water from its higher concentration to lower concentration. That also will take place. Means you can see here water has started to move inside. That is due to this osmotic pressure. The presence if at any time the cell wall is unable to bear the Turgor pressure, it ruptures and the cell contents burst out. That is what I have explained you. If the Turgor pressure exceeds the wall pressure, in that case what will happen? The cell will burst. This is what happens when certain fruits and vegetables sometimes they burst. You may have seen certain fruits and vegetables, they burst. When turgid, the cell is in somewhat balanced state. When the cell is fully filled with water, the wall pressure and the Turgor pressure is equal. At that time it is a balanced state. No more water is entering or leaving. At this condition, when the cell is fully turgid, at this time no water is entering, neither no amount of water is leaving it. Its Turgor pressure counterbalances the wall pressure. Already that I have told you, the Turgor pressure will counterbalance the wall pressure. Now the plasmolysis and deplasmolysis. When a living cell such as the cell of a leaf of an aquatic plant is placed in fresh water, okay, suppose any leaf you take and it is kept, any leaf means the aquatic plant we can choose, okay, a leaf is chosen and it is placed in fresh water. It remains fully distended condition. It will remain in distended condition. Its plasma membrane remains in close contact with the cell wall and it will press against it. Means the cells will remain fully turgid. Now, what we are supposed to do? We are supposed to take out that plant cell or take out that leaf, and we are supposed to place it in a solution containing 5% salt. Means we are supposed to transfer it to a 5% salt solution for a few minutes. So what we will observe? We will observe that since the concentration of water outside is more, water from the cell inside will start to move outside by the process of exosmosis. The cytoplasm will start to shrink, and when the cytoplasm will shrink, one thing we have to keep in mind: the cell membrane will, when the cytoplasm will shrink, the cell membrane will also shrink, whereas the cell wall will remain intact. The cell wall there will be no change in the cell wall, but the cell content will shrink and it will come to a corner. So the cytoplasm will shrink, and the plasma membrane will withdraw from the cell wall, and we will also see the plasma membrane also withdraws itself along with the cellular components to a corner, whereas there will be no effect on the cell wall. The cytoplasm will shrink, the plasma membrane along with the plasma membrane, and the cell wall will remain as such. This shrinkage from the cell wall is called plasmolysis. So I have told you, as the cell is kept in a hypertonic solution of 5%, 5% salt or sugar solution, when it is kept, water will start to move from its height, means from inside the cell to outside due to exhaustion. Due to this loss of water, what will happen? The cellular content will shrink, and along with the plasma membrane, okay, we are leaving the cell wall, and this condition is called as plasmolysis. The cell is in a state, this state, when the cell enters this state, it is called as limp or flaccid. Such kind of cell is called limp or flaccid, and the condition is termed as flaccidity. This is flaccidity, and the condition is termed as flaccidity. If this flaccid or plasmolyzed cell is returned to water before it is dead, now we have to see one thing, that if this flaccid cell or plasmolyzed cell is again taken out from that 5% solution and brought to normal or hypotonic solution, what will happen? It's returned to water before it gets dead. Its protoplasm, again they regain the water, it will swell up, and it will start to touch the cell. This condition will again, it will attain, okay, since it regains the water, the cell membrane will start to, it will come in contact with the cell wall, okay. So when the cell comes from again regains from its plasmolyzed condition, or from its flaccid condition, that is termed as deplasmolysis, okay. Plasmolysis and deplasmolysis, that I have explained you, okay. This protoplasm again swells up against the cell wall, and this is called as deplasmolysis. Now, what is the significance of turgidity? Turgidity provides rigidness to soft tissues. Okay, we will see the significance of turgidity. First thing is, all the cells of the plant, they remain turgid. Due to this turgidity only, when they are rigid, when they are turgid, okay, the leaves are expanded, the plant is erect. When all the cells are fully turgid with water, when they lose water, what happens? They will wilt down, or the leaves will droop down. When there is not enough water in the leaf, it wilts. You may have seen, suppose you don't water a plant, a potted plant you take, you don't water it for some time, you find the plant starts to wilt. What are the signs and symptoms of wilting? You will find the stem will bend, isn't it? The leaves will droop down, isn't it? They will not remain in their expanded form. So there is not enough water in the leaf, it wilts, its petiole and lamina becomes loose, and the leaves droop down. Wilting of leaves is usually noticed when the plant is exposed to hot afternoon sun. Usually we have seen during the summer season, when certain plants, herbaceous plants, when they are during the daytime, under the scorching heat of the sun, okay, what happens? They wilt. Okay, the stem droops down, the stem bends, the leaves droop down. But in the evening again, or during the night sun, the temperature is so much that there is too much loss of water that takes place from the aerial parts of the plant. Okay, or in other terms, this loss of water from the aerial parts of the plant is termed as transpiration. Okay, so when this transpiration, too much of transpiration takes place, so there is loss of water in the plant. And on the other side, water is continuously drawn through the roots from the soil. Okay, so what actually happens? The rate of transpiration exceeds than the rate of roots. Okay, so what happens? In order to cope up that condition, the plants wilt, or the leaves, they droop down. Exposed to at this time, the amount of water lost during transpiration is more than the water absorbed through the roots. In the evening, when the rate of transpiration falls down, the good quantity of water is reabsorbed, it is replenished, the loss of water during the daytime. During the daytime, whatever water has been lost, that is replenished, and the leaf again become fully erect or distended. The leaf cells restore water and becomes turgid, thereby making the leaves stand out. The leaves again they expand. Turgor pressure also helps to push through the hard ground. Okay, we may have seen when a seed germinates or mushroom, when they come out of the soil or the sand, or the seeds when they germinate. Okay, we have also seen the roots penetrate through the soil. Okay, actually the tips are rigid, the tips of the roots are rigid, and it is due to this turgor pressure that they are able to penetrate deep inside the soil. Plants growing on the walls cause cracks. Some of the plants you may have seen that you find growing on the walls of monuments, various kinds of buildings on walls. They exert so much of turgor pressure that they may break the monuments or the buildings or the walls. So due to the higher turgor pressure, then turgor movement. This is seen in various kind of sensitive plants you may have seen, like touch me not plant. The botanical name of touch me not plant is Mimosa pudica. So you may have seen in Hindi it is called as chui mui. When you touch a chui mui plant, what happens? Or touch me not plant, the leaves droops or it gets folded. Okay, actually this plant is sensitive to touch. When you touch the plant, that part of the plant loses its turgidity. Okay, the water from that part, making the cells flaccid, due to which the leaves get folded. But after some time, when you leave the plant undisturbed for some time, again they regain the water, or the water is replenished and they become expanded. It is seen in some sensitive plants, Mimosa pudica. This type of movement is also seen in some insectivorous plants. You may have seen quick movement in insectivorous plants. They are also sensitive to touch, and they show such kind of movement. If one leaf of a plant of this touch me not plant is touched, even lightly, if the leaflet folds up slowly, the leaves recover and again stands erect. Here, the stimulus of touch leads to loss of turgor. So when we are touching, it will lose the water. The cells will lose their water. At the base of so what will happen? The water from the cells of the leaves, it will leave and it will move towards the base of the leaflet and the base of the petiole, and it will start to move towards the base of the petiole, which is called as the pulvinus. Okay, the base of the leaflets, which is called as the pulvinus, where it gets stored for some time. But when it is again left undisturbed for some time, it again comes back to the cells of the leaves. There are some other examples of this turgor movement, like sunflower. You might have seen the sunflower always faces the sun. That is why it has got the name sunflower. Okay, sunflower moves towards the sun. That is also a kind of turgor movement. You may have seen sleep movement in various kind of flowers. So there are flowers that will close during the night time, but again they will open during the daytime. Okay, like the lotus also you will find that closes during the night time but opens in the daytime. Okay, so that are called as the sleep movements of the leaves as well as plants or in various plants. Next is turgor in opening and closing of stomata. We all know stomata are very important. They are found on the aerial parts of the plant under the leaves. Okay, these are the pores through which exchange of gases takes place. Not only exchange of gases, but these pores are also very much helpful in the transpirational process, that is loss of water through the aerial parts of the plant. So this opening and this stomata or the stomatal aperture, when we are studying, we have to know the structure. They are having two kidney shaped cells which are called as guard cells. They are fully filled with water, and the turgidity and flaccidity of this guard cells enables the stomatal aperture to close and open. When guard cells are turgid, when they are fully filled with water, they are fully distended. Their walls get distended and they move away. Okay, so these kidney shaped cells, guard cells, when they are fully filled with water, okay, but the inner cells, they are distended, they become apart from each other, making the pores open. This makes open when the guard cells are flaccid. On the contrary, when the guard cell loses its water, it will become flaccid or loose. As soon as it will become flaccid or loose, it will close the stomatal aperture. When guard cells are flaccid, their walls collapse. Thus, stomatal opening is closed. Turgor pressure or turgidity in root cells builds up the root pressure. For this, we can take a well-watered plant. We can understand this with the help of example, with an experiment. So what are we supposed to do? We have to take a well-watered plant, and then the well-watered potted plant is cut. This little amount of stem is left from the base. It is not cut just from the base, just above the base. It is cut. When a well-watered plant is cut a few centimeters above the soil, what happens? Water will start to ooze out from that part. Immediately, a glass tubing is fixed by means of a rubber connection. So you like this, you can see the stem is cut just above the base, and a glass tubing is attached to it with the help of a rubber tubing. This glass tubing is connected to a mercury containing manometer. Okay, so what happens? Immediately the glass tubing is fixed by means of a rubber connection. Water will start coming out through the cut end of the stem, and it will start to rise up in the glass tubing. What will happen? From the cut end, water will start to ooze out. As it will ooze out, it will start to move inside the glass tubing. It will fill the glass tubing, and it will, you can see here the level is, mercury level is similar both the sides on both the arm. But what will happen due to the pressure generated by the oozing liquid? What will happen? The mercury will be pushed, and you will find a rise in the manometer, in the mercury level in the manometer. The rising water raises the mercury filled in connected manometer. The upward flow of water is due to the heavy pressure from the roots. So this pressure, you can see the pressure through which the water is oozing out. It is through this pressure only upward movement of water or cell sap takes place, and this is only termed as root pressure. That it can be defined as the pressure which is generated in the roots due to continuous inward movement of water through cell to cell osmosis. Once again, I will tell you the definition. The pressure generated in the roots due to continuous inward movement of water through cell to cell osmosis. It is termed as root pressure. Loss of water. Now, you may have seen sometimes when any part of a plant is cut, or when you pluck a leaf, you find the liquid, sometimes latex or sometimes transparent cellular fluid that starts to ooze out, cell sap that starts to ooze out. That is termed as bleeding. Okay, loss of water through the cut stem of the plant is called as bleeding. This is also a very important term for in the syllabus that you need to know. Now let us start to move. Let us move ahead with our topic itself. That is, we will be understanding now some practical utility or practical application of plasmolysis. So we will see the practical usage of plasmolysis. You may have seen the meats or the pickles, when they are made, large amount of salt is added to it. So salting or adding of salt is a method of killing the bacteria. How does the bacteria get killed? Is the salt poison? Yes, actually it's not the poison, but the salt makes a hypertonic solution, due to which the cellular contents from the bacteria comes out through exosmosis, and this renders the bacteria harmless. Okay, salting of meat and pickles is a method of killing bacteria by plasmolysis. The bacterial cells becomes plasmolysed. On the same thing is that the cells of the meat, they lose water. When water will not be there, no bacterial action can take place. In the same way, weeds can be killed in a playground. So in order to kill the weeds, the those plants which are not needed, unwanted plants, are called as weeds. For the removal of these weeds, uh, salt is sprinkled. So what happens? Again, the cell becomes plasmolized. When the cell will become plasmolized, when it will lose its water, so all the physiological functions of the plant cell will stop. When the physiological functions will stop, naturally the plant will die, or the weed will die. Excessive salt is sprinkled around the base of the plants or the weeds. High concentration of sugar is added in jams, jellies, and squash, various kinds of drinks. This also is used to plasmolyze the bacteria and the fungi that will attack on the jams and jellies or those eatables. Then fish and vegetables, these are dried to increase their shelf life. The fishes and the vegetables, these are dried and they are salted. That also is brought about in order to prevent the fish and vegetables from the spoilage by bacteria and fungi. Then uh, plants growing in the walls. Plants growing in the walls, in the foundation, you may have seen when the plants, they grow in the walls of monuments and buildings. Okay, they will grow. Okay, not only they will grow, but due to the turbo pressure, they can make cracks, deep cracks, and can destroy the buildings. They can destroy the building. So such plants can be prevented from growing. Such plants can be prevented from growing by the addition of salts. Absorption of water. Let us see how absorption of water takes place by the root. So it takes place by means of, you all know, it is the roots are provided with tiny or fine root hairs. So the root hairs, they absorb water. So it takes place by means of the root hair. Root hair, the cells of the root hair, they contain cell sap, which is at a higher concentration. The concentration means they are having higher concentration of salts. Large amount of solute is present in that, due to which what will happen? Water will start to move from the soil to the inside of the root cells. So the concentration is higher as compared to the outside soil water. So there is a difference. This difference sets off osmosis. Due to this osmosis, which kind of osmosis? Endosmosis will take place. Water from outside will start to diffuse inside the root hairs. Water continues to pass through the adjoining cells. From one cell to the other cell, the movement of water will start to take place. Okay, and first of all from the root hairs, okay, then to the endodermis, from the endodermis to the pericycle, and at last to the xylem cells, and then the tracheids, it will move, and finally water enters the xylem vessels, from where it will be transported to the part wherever it is required. The turgidity acquired by the cells helps to push the water upward through the xylem vessels. So the turgidity acquired by the cell, that will be helpful in pushing the water upward. How does the absorption of mineral will take place? We all know that cell wall is freely permeable, but cell membrane is selectively permeable. It will allow the movement of water through it, but not the solute particles through it. So as minerals being solid, or minerals are usually absorbed in the form of ions. Okay, and that will involve active transport. Active transport was explained to you in the last class. So it involves active transport by the cells. So external, they will require energy in the form of ATP. Okay, for transferring these mineral elements in the form of ions. Minerals are absorbed as ions rather than in the form of salts. Means they are absorbed in salts, but more readily they are absorbed in the form of ions. So these ions get dissolved in water, and along with water they move when the path is same, that is from the root hairs to the endodermis cell, from the endodermis cell to the xylem, and so on, and they will move to the parts of the plant wherever it is required. The dilute solution of water and mineral salts is absorbed from the soil by the roots. It is used for food manufacture in the leaf. So these minerals, we know water is very essential for the process of photosynthesis. Along with that, along with water, minerals are also required. Okay, since they minerals play a very important role and they are very important components or constituents of various organelles of the plant cell. The upward flow of minerals occurs through the xylem. As in water, the movement of upward movement of water will take place with the help of xylem. In the same way, upward movement of minerals will also take place by xylem itself, since these minerals in the form of ions are dissolved in the water. What next topic is ascent of sap. What is ascent of sap? Movement of upward movement of water against the gravitational force is termed as ascent of sap. You may have seen very tall plants like that of a coconut plant. Okay, it has to they are massively very long or very tall. Okay, and the water has to be transported from the root to the top of the tree against the gravitational force. How does this take place? There are so many forces which come together to bring about this upward movement of water, that is called as ascent of sap. Upward movement of water and minerals from the xylem of roots through the stem and leaves of veins to the top of the plant is termed as ascent of sap. Upward conduction takes place through the lumen of the xylem vessels and tracheids. So this xylem vessels are in the form of lumen. So through this lumen itself, the movement of water will take place from the root to the upper part of the plant. So there are some causative agents. I've told you now, this movement or this ascent of sap results with a combination of various types of forces. So the causative forces of ascent of sap, the very first force is capillary force. So we know very in capillary, you might have seen a capillary. Inside of it is having a very narrow lumen or somewhat vacuum. Whenever that capillary is dipped in a liquid, it takes the liquid, or vacuum is filled by the liquid. Okay, so the narrow diameter of the xylem acts as a capillary and causes the water from the lower level to rise to fill up the vacuum created by the loss of water. How is this vacuum created in the lumen of the xylem? Actually, there is continuous loss of water from the aerial parts of the plant by the process of transpiration. This loss of water results into formation of vacuum in the lumen of xylem. Okay, due to which this vacuum space is filled up by the capillary force. Due to transpiration, narrower or the diameter of the tube, means narrow is the diameter or the lumen of the xylem, greater will be the height of the water rising in it, exerting a force, and this force is termed as capillary force. The next is root pressure. Root pressure may be enough when we are talking about root pressure. It may be enough for herbaceous plant for the upward movement of the cell set of the plant or water for herbaceous plant, but not for very tall plants. Okay, but not for very tall plants. This root pressure is applicable. The magnitude of root pressure is does not exceeds more than 2 to 3 atmosphere, whereas a pressure of about 16 to 20 atmosphere is required to raise the water from the roots to the top of the plant. Okay, so this root pressure cannot be, but it is helpful in the upward movement of water in the case of herbaceous plant which are having short height. It also shows seasonal fluctuation. Seasonal fluctuation when we are talking about, suppose rainy season, when the atmosphere is having large amount of water vapor and the soil also has a large content of water in it. So it shows seasonal fluctuation being highest. It will be highest in the root pressure will be highest in the spring and it will be lowest in the summer. Why will it be lowest in the summer? Because there is very less amount of water in the soil as well as in the air. Now, transpirational pull and cohesive adhesion tension. What is cohesion? Cohesion means the attraction between two similar molecules, that is cohesion. Whereas adhesion means it has come from the word adhere, means when we are adhering two different types of molecules when they are attached to each other, that is called as adhesion. So cohesive and adhesion tension is formed, as well as the transpirational. I have explained you, when transpiration takes place, loss of water from the aerial parts of the plant will take place. Naturally it will create a vacuum in the xylem tubes. Due to this vacuum, what will happen? A transpirational pull is created, due to which upward movement of water will start to take place. That will result into ascent of sap. Water is lost from the leaves by transpiration. More water molecules are pulled up due to this tendency of water to the of water molecules to remain joined. There is a tendency in the water molecules to remain joined. This tendency of water to remain joined or to form a continuous column is called as cohesion. This produces a continuous column of water through the stem. This is through the cohesive force. Now adhesion means adhesion causes the water molecules to stick to the wall of the xylem, lumen of the xylem, or the surface of the cells. More molecules of water is drawn from below. So adhesion and cohesion all together work together and bring about upward movement of water. When the leaf cells lose water during transpiration, the suction force is provided by the leaves. It is very important for tall trees like coconut, okay, pines, which do not have enough root pressure. Since when we talk about root pressure is not sufficient or enough to transfer the water to such an enormous height. Okay, in such case, this transpirational pull and cohesion, adhesion, tension displays a very important role to bring about ascent of sap. So let us see, there are some activities also that we need to understand. This activity is there in your course in order to show that root absorbs water. And the second activity is to show that conduction of water or the ascent of sap takes place through xylem tissues. Experiment to show roots absorb water. For this experiment, what we are supposed to do, you are supposed to take two test tubes, name them A and B. Okay, after taking these two test tubes A and B, we are supposed to fill both the test tubes with water. Equal amount of water is filled in both the test tubes, and above that few drops of oil is put. Okay, we have dropped few drops of oil in order to prevent surface evaporation. In order to prevent surface evaporation, few drops of oil is immersed. Okay, it is dropped into the test tube. Now after that, in test tube A, rooted plant is taken, a plant which is provided with a root that is immersed into this first test tube A, whereas the second test tube is left without water, without this plant. Okay, since both the condition here, we will find that it is having water, it is having oil, the amount of water is also equal, but one condition is missing, and that is the presence of plant in B. So this will act as a control. Whenever we are conducting any experiment, there is a control experiment. So this will be a control setup. Now we have to leave the plant as such for some time. So when we will leave it for some time, we will find that there is after some time we will find that there is decrease in the level of water in test tube A. Why is there a decrease in the level of water in test tube A? Because the plants, why we have used oil, so no surface evaporation is going to take place. Okay, but the roots of the plant will continuously draw water from. Why will it draw water? Because there is loss of water from the leaves, from the aerial parts of the plant through the stomata by the process of transpiration. Okay, due to which a vacuum is created. Okay, the transpirational pull is created. Due to this transpirational pull, large amount of water will start to get absorbed by the roots. Okay, and a continuous channel of water will be formed inside the xylem lumen. Okay, now what will happen? We will find there is a decrease in the level of water. But what will happen to the second test tube? The second test tube, we will find there is no decrease in the level of water. Why there is no decrease in water? Because water has not been absorbed by the roots, and no surface evaporation is taking place since we have used oil here. Okay, so this proves that roots absorb water. Roots are the basic organs for absorption of water. Let us come to the next experiment. The next experiment is to show that water is conducted through xylem only. For this, what we are supposed to do, we are supposed to take a beaker. In a beaker, we are supposed to take water, which is where eosin, it's a kind of dye, is dissolved, or a beaker containing eosin solution. We have to take this. It is red in, this eosin is red in color. After that, we are supposed to take an uprooted plant, and we are supposed to immerse that uprooted plant into the beaker. We have to put the whole setup like this, and we are supposed to keep it for some time, undisturbed for some time. What will happen? I've told you in the previous experiment that roots will absorb water. In the same way, roots will absorb water. What kind of water will they absorb? The water containing eosin dye. It will absorb that water. Okay, since water transpiration will take place, upward conduction of water will take place, so the xylem uh, will start to absorb the water containing eosin. Since we have used a red color water, the xylem will also attain a dye. It will get the dye. It will attain a red color dye. After some time, what we are supposed to do, we are supposed to cut the transverse section of various parts of the same plant that we have immersed in the beaker. So we will find when we have cut the transverse section of the root part, a transverse section of root, we will find the centrally placed xylem, the xylem position. Okay, the part which is the central part that is formed of xylem, it is turned red. From where has it got the red color? Due to the conduction of this eosin solution, it has attained red color. So that this will show the position of xylem. Moving further, when we will cut the transverse section of stem, we will find the xylem is radially arranged in this way, and we will find the xylem cells have attained a red color. It will the water will not stop here only. It will move in the leaves also, and when we will cut the transverse section of the leaf, the mid-rib portion containing the xylem cells, that also will attain red color, and they will get the same color of the dye. They will turn red into color. So by this experiment, it is proved that water is conducted through the xylem. The xylem that is present in the roots, in the branches, and in the leaves also. So this was all about the chapter. You have to go through this these topics that I have explained to you, and you have to see various terms which are given in your book as well, like cohesion, cohesive forces, adhesion, adhesive forces. All these terms you are supposed to go through. There is some homework also that I have given you right now also, and some of the homework that I'll be publishing on the website, or school's website, that you are supposed to do. Notes that I will provide, that you are not supposed to write in your copy. That is only for reference. Okay, but the question answers that I will be giving you, that only you are supposed to write in your copy. Okay, you can refer the notes that I have provided for writing the answers, as well as you can refer the book for writing the answers of those questions. Let us move to the PowerPoint presentation that I have prepared for the day for the same topic. Chapter 4. Absorption by roots and rise of water up to xylem. So we are continuing this chapter. This is a continuation of the chapter. Turgidity and flaccidity. Every living plant cell, directly or indirectly, is in contact with fluids. The root hairs in particular are surrounded by soil water. All such cells are subjected to osmosis, and the water continues to enter as long as the cell sap is more concentrated than the surrounding fluids. Means from the surrounding fluids, water will start to enter inside the cell by endosmosis. When a cell reaches a state where it cannot accommodate any more water, it is fully distended and it is called turgid, and the condition is called as turgidity. The cell is turgid, whereas the condition is turgidity. When a cell is turgid, its water is stretched under pressure from inside. In its turn, it presses the cell contents towards the center of the cell. When a cell reaches a state where it cannot accommodate any more water, it is fully distended. It is called turgidity. When the cell is turgid, its wall is stretched under pressure from inside towards the center of the cell. The pressure of the cell contents on the cell wall is called turgor pressure. The pressure exerted by the cell wall on the cell content is called wall pressure. It is just an opposite to turgor pressure. If any time the cell wall is unable to bear the turgor pressure, it ruptures and the cell contents burst out. This is what happens when certain fruits and vegetables sometimes burst. When turgid, the cell is in somewhat balanced state. No more water is entering or leaving it. Its turgor pressure counterbalances the wall pressure. This is the diagram that I have drawn on the board also, showing osmotic pressure, turgor pressure, wall pressure. A plant cell showing all these three, which is represented all together. Plasmolysis and flaccidity. When a living cell, such as the cell of a leaf of an aquatic plant, is placed in fresh water, it remains in a fully distended condition. Its plasma membrane remains in close contact with the cell wall and presses against it. If this plant cell is now kept in 5% salt solution for few minutes, it will lose its distended appearance. It will start to lose the water due to exosmosis. The cytoplasm will shrink and the plasma membrane will withdraw from the cell wall. This shrinkage from the cell wall is called plasmolysis. So the shrinkage of this plasma membrane from the cell wall is termed as plasmolysis. The cells of this state are said to be limp or flaccid. This condition is called flaccidity. If this flaccid cell or this plasmolyzed cell is again placed in water, that is in a hypotonic solution, before it is dead, its protoplasm regains the water, it will swell up against the cell wall. This reversal of plasmolysis is called deplasmolysis. You have to keep in mind the definition. What is the meaning of plasmolysis? What is the meaning of deplasmolysis? What is the meaning of flaccidity? What is flaccid? All these things you need to keep in mind. You can see here two cells, cells in water in normal turgid condition. You can see all the cells are in turgid condition. In diagram B, in the picture B, you will find when the same cell is kept in 5% salt solution, protoplasm will shrink. It will leave the cell wall, and it will shrunk to a point. You can see here the diagrams depicting C, D, E to F, where it's a diagrammatic representation of sequence of plasmolysis, where you can find in C you will find a turgid cell which is kept in a hypertonic solution, and the cellular content the protoplasm starts to shrink, and in F you will find it completely shrunk. It's a plasmolysed cell. Okay, and the liquid which is filled inside is the hypertonic solution. Significance of turgidity. Turgidity provides rigidity to soft tissues such as the leaves. When there is not enough water in a leaf, it wilts. Its petiole and lamina becomes loose and the leaves droops down. Wilting of leaves is usually noticed when a plant is exposed to hot afternoon sun. At this time, the amount of water lost during transpiration is more than the water absorbed through the roots. In the evening, when the rate of transpiration falls down, the quantity of water absorbed replenishes the loss of water during the daytime. The leaf cell restores water and becomes turgid, thereby making the leaves stand out. It becomes expanded again. Turgor pressure helps to push through the hard ground. Roots are able to penetrate through the soil since their tips are rigid. Plants growing on the walls cause cracks in walls and concrete. Even the concrete floors, they can cause crack due to their higher turgor pressure. Turgor movement. This is seen in some sensitive plants like Mimosa pudica, commonly it is called as touch me not plant, and also in some insectivorous plants. If one leaf is touched even lightly, the leaflets folds up. Slowly the leaves recover and stand erect when left undisturbed. Here the stimulus of touch leads to the loss of turgor at the base of the leaflets and the base of the petioles called pulvinus. Let us see some other examples. Movement of certain plants like the sunflower towards the sun. Sleep movements of flower and leaves of certain plants. Turgor movement in the opening and closing of stomata. The opening and closing of stomata is controlled by guard cell. When the guard cell are fully turgid, their walls are distended, they will move away and the stomatal aperture is open. This will make it open. When guard cells are flaccid, their walls will collapse and the stomatal opening is closed. Now turgor pressure, turgidity in the root cells cells builds up root pressure. This already I have explained this experiment. Okay, where a plant which is cut, the upper part of the plant is cut and it is introduced with a glass tubing with the help of rubber, you can see here, and it is attached to a manometer. This is the manometer, and you will find due to the pressure of the cell sap which oozes out from the cut part of the stem, it will push the mercury and the mercury level will rise in the manometer. This will show the turgor pressure experiment to prove turgor pressure. When a well-watered plant is cut a few centimeters above the soil, immediately a glass tubing is fixed by means of a rubber connection. Water will start coming out of the cut end of the stem and rise up in the glass tubing. Rising water rises the mercury filled in the connected manometer. The upward flow of water is due to heavy pressure from the roots, and this heavy pressure is only called as the root pressure. Let's see the definition of root pressure. The pressure generated in the root due to continuous inward movement of water through cell to cell osmosis. Loss of water through cut stem is called bleeding. When any part of the plant is cut, we will find the cell sap oozing out. Sometimes the latex oozes out. That is termed as bleeding. Practical application of plasmolysis. Salting of meat and pickles is a method of killing bacteria by plasmolysis. Weeds can be killed in a playground by sprinkling excessive salts around their base. High concentration of sugar in jams, jellies, drinks, etc. protects them from bacteria and fungi. Fish and vegetables are dried and salted to protect them from the spoilage. Plants growing in the walls and foundation of monuments, buildings, bridges, etc., they are also killed by the same way, means by addition of salt. Absorption of water, how does it take place? It takes place by the means of root hairs. Root hairs contain cell sap which has a higher concentration of salt as compared to the outside soil water. This difference sets off osmosis. Water from the outside diffuses into the root hair. So this is the diagram how the movement of water takes place from the roots. You can see here these are the soil particles through the root hair. This is the channel through which the water particles will move from the root hair, that is through the root hair to the epidermis, then through the cortical cells it will move through the endodermis and the pericycle into the xylem tissues. This is a cross section of the part of root showing the passage of water. Water continues to pass to adjoining cells from the cell bearing root hairs. Finally, water enters the xylem vessels. The turgidity acquired by the cell helps to push the water upward through the xylem vessels. Absorption of minerals. How does it take place? It involves active transport method for its absorption. Minerals are absorbed as ions rather than as salts. The dilute solution of water and mineral salt is absorbed from the soil by the roots. It is used for food manufacture in the leaves. The upward flow of minerals occur through the xylem. Ascent of sap. The upward movement of water and minerals from the xylem of root through the stem and leaf veins to the top of the plant is called ascent of sap. This is the definition of ascent of sap. The upward conduction takes place through the lumen of the xylem vessels and tracheids. What are the causative forces which brings about ascent of sap? Capillary force. Narrow diameter of the xylem vessels causes water from the lower level to rise to fill up the vacuum created by the loss of water due to transpiration. Narrower the diameter of the tube, greater will be the height of water rising in it, exerting a force which is called as capillary force. Root pressure. The root pressure may be enough for herbaceous plant, but it is not enough for the rise of water in case of tall plants. Because the magnitude of root pressure, it does not exceeds more than 2 to 3 atmosphere. On the contrary, a pressure of about 16 to 20 atmosphere is required to raise water up to the top of the heighted trees. It also shows seasonal fluctuations being highest in the spring and lowest in the summers. Transpirational pull and cohesion adhesion tension. Water is lost from the leaves by transpiration. Already it has been explained how does when transpiration takes place. There is continuous loss of water in the form of water vapor from the aerial parts of the plant. More water molecules will be pulled up due to this tendency of water molecules to remain joined, since water has a tendency to join together by the force which is called as cohesive force or cohesion in order to make a continuous channel. Along with that, this produces a continuous column of water through the stem. Along with that, adhesion also causes the water to stick to the surface of cells. More molecules of water is drawn from below. In this way, both of these forces will act together and more molecules of water will be drawn from below, that is from the soil. When the leaf cells lose water during transpiration, the suction force is provided by the leaves. It is very important for tall trees like pines, coconut, etc., which do not have enough root pressure. Now we'll be seeing the activity. Already it has been explained. The activity to show that root absorbs water. Where you can see here test tubes are taken. Okay, one is the control and the other one is filled with water, and oil is also dropped into it, and we will find initial level of the water and final level we will find it over here. Why is there a drop in the level of water? Due to absorption of water by the roots. So this experiment will show that root absorb water. The next experiment, that is the activity to show that upward conduction of water takes place through xylem, where an uprooted plant is placed in a beaker containing eosin solution, where the roots will start to absorb water. As we know, the upward conduction of water will take place by the xylem cells. We will find the xylem also attaining the color of the dye. And when the transverse section of the various parts of the plant like the roots, the stem and the leaf is carried out, it is seen the central xylem portion acquires red color dye, that is eosin. This proves that the upward conduction of water takes place through the xylem. So this was all about the chapter. Now please note down some homework questions that I'll be giving you. You are supposed to write these homework questions in your copy. The very first question is, what is the utility of plasmolysis? Second question, give differences between turgidity and flaccidity. Third question, define turgor pressure, root pressure. Fourth question, how does turgor pressure help in closing and opening of stomata? I will repeat all the questions once again. The very first question is, what is the utility of plasmolysis? Second question, give differences between turgidity and flaccidity. Third question, define turgor pressure, root pressure. Fourth question, how does turgor pressure help in closing and opening of stomata? So this was all about for today. Please write down the questions in your copy and see whatever I have explained. Read it from the book also. Refer to the books or the note that I will be giving to you. Thank you and good day.